Let's move on to part 3 of Unit 7. We're going to talk about some unusual antigens within the Rh blood group system. And there's many weirdos in the Rh system, but you're only going to be responsible for the ones presented in this lecture. So let's talk about compound antigens. A compound antigen are epitopes which occur due to the presence of two Rh genes on the same chromosome, meaning they are in the cis position. Gene products include not only the products of the single gene, but also a combined gene that is also antigenic. For example, the most well-known is little f, and you will see this on your antigram when you're doing your screens and you're doing panels. The F antigen occurs when little c and little e are found in the cis position. The gene makes little c and little e, but when they're together on the same chromosome, for some reason this creates its own antigen known as little f. And this only occurs again when the little c and little e are in the cis position. Little f will not be present in the trans position. Antibodies to compound antigens are rare. If they were identified, finding negative units would be done using the routine Rh typing series. So you would type your donors for big C, big E, little e, and D. For anti-F, if you find a donor negative for little c or little e, this would ensure the unit is negative for F. So again, little c and little e must be present on the same site. So if you're little c negative or little e negative, you will not be capable of having F. There are other compound antigens, but you're responsible for describing the F antigen and finding compatible donors. And how you're going to find compatible donors, again, would be the unit being little c negative or little e negative. G is a combination antigen found on cells containing either D or C antigens. It doesn't have to be both. Either D or big C. Given the high frequency of D, it's not surprising that most people express G. The main issue is that G individuals can form an antibody to G that will react against either D or C positive cells and cannot be separated into two different antibodies. It's similar to anti-A, B, which reacts with both A and B cells. You cannot separate out the specificity. It's anti-AB, just like G is anti-CD. It's if either the C or the D antigen is present, it will react. Ruling out separate anti-C and anti-D takes some work. So you attempt to absorb out, get rid of, through a procedure, anti-big C. And just briefly, you take C positive reagent cells, you add the patient's serum, and you allow it to incubate, and then you harvest the serum. And what you're hoping is that the anti-C will have been absorbed onto the reagent cell and won't be present. So then you test the absorbed serum against a C positive and a D positive. If this is truly an anti-G, the serum will react with both, because remember, it's not an anti-big C separate anti-D, it's an anti-CD. If the patient is D positive, transfuse them with D negative, and most D negatives are little r, so it won't be a problem finding a blood for D negative patients with anti-G. D deletions are very, very rare, and at the MLT level, I just want you to be able to describe what these are. These individuals inherit an Rh gene complex lacking alleles. It may be at the E or the C. Must be homozygous for this rare deletion to be detected. So there's no reaction when red cells are tested with anti-E, little e, big C, or little c. I want to say that again. They're negative with all Rh typing series. You say that can't be possible because there are 5 alleles, and I know genetics, and they need to be inherited. So this is a very, very rare genotype. It requires transfusion of other D deletion red cells because these individuals may produce antibodies with either single or separate specificity. So theoretically, they can make an anti-big E, an anti-little e, an anti-big C, an anti-little c, and you're not going to find blood anywhere except for a person who also has this rare deletion genotype. So it's written as either D dash dash or dash D dash. I love to say that. There's also one called dot D dot, and that one has a weaker expression of the D antigen than a dash D dash. So again, D deletions are very rare. I want you to know they're negative for all Rh antigens except D, and they can form really, really complex antibodies, which will result in the transfusion of rare donor blood. And one of the most rare genotypes is the Rh null. They have no Rh antigens, none. You write the genotype dash 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 slash dash dash dash, no Rh antigens. And Rh antigens are an integral part of the red cell membrane. So the lack of these antigens will cause the red cell membrane to appear abnormal, leading to stomatocytosis and hemolytic anemia. And I want you to know that. Those are two characteristics of the Rh null genotype. There's two Rh null phenotypes. The regulator type, the gene is inherited but not expressed. And then the amorph type, where the RhD gene is absent, totally absent. So there's no expression of the Rh gene. Complex antibodies may be produced requiring the use of rare, autologous, or compatible blood. And the best place to look for compatible blood in the general population is look at the siblings, hoping you have some. The LW antigen was discovered at the same time as the Rh or rhesus antigen. The LW was detected on the cells of rhesus monkeys and human RBCs in the same proportion as the D antigen. They initially thought this was the same antigen, but eventually they discovered some distinct differences. It's named LW in honor of Landsteiner and Wiener. There are rare individuals that lack LW, yet have normal Rh antigens. Individuals can form allo anti-LW, and one of the key characteristics of this antibody is that it will react more strongly with D pos than D neg cells. Keep in mind when a D positive individual appears to have anti-D. In my entire career, I've only seen one, and it was really, really cool because the person appeared to be D positive, yet they had this really potent anti-D. And so, again, a lot of research done, and I think we eventually sent it off to a reference lab for confirmation. CW is a variant Rh antigen. It's very, very low frequency, meaning it's found in a small percent of the population. In this case, 1 to 2% of the whites and rare in blacks. Most individuals who are big C positive are also CW positive. Antibodies to these antigens can be naturally occurring and may play a role in hemolytic disease of the fetus and newborn or hemolytic transfusion reactions. So again, we're going to talk about frequencies when we get into identification of alloantibodies, but a low frequency antigen is one that is absent in the majority of the population. So if you have an antibody, finding donor blood is very, very, very easy because most people lack the antigen. High frequency, unfortunately, is the opposite. It's found on almost everybody, so chances of finding compatible blood are very difficult. Let's talk in general about Rh antibodies, and this will be repeated in other lectures as well, because the more times you hear this information, again, I've said this before, the better it will stick. Anti-big E is the most frequently encountered Rh antibody, followed by anti-little c. I want you to know that. Anti-big C is rare as a single antibody. It's usually found with others. Anti-little e is rarely encountered. It's only 2% of the population is antigen negative. And it's kind of a good news, bad news, because if you do have an individual with anti-little e, only 1 out of 100 donors will be negative for the antigen. So if you need more than one unit, you're going to be screening a bunch of blood. Detectable antibody persists for many years and sometimes for life. The antibody may fall below the level of detectability. What that means is you do the antibody screen, it's negative, and then you transfuse the person, and due to re-exposure, this will cause a secondary immune response. So this is why when we get into antibody identification, the patient's history is so crucial. If a patient is ever labeled as having an antibody, an immune antibody, for the rest of their life, they must get antigen-negative blood. Anti-D may react more strongly with R2-R2 cells than R1-R1 due to the higher density of the D antigen on the cells. Now you're at a point, I want you to look at your screen cells and look at the Rh genotype. If you're using a 3-cell screen, you will have an R1-R1, R2-R2, little r. This way we have all the antigens in the Rh system. Some manufacturers make a 2-cell screen, which is an R1-R1 and an R2-R2. So this is, again, I'm teaching you something, the language of blood bank. We use it all the time. And again, knowing that anti-Ds react more strongly with R2-R2 than R1-R1 may explain the varying strengths of reactions, which is really important when we go into working up and doing an antibody panel. This chart is from your textbook, and the clinical considerations first, let's talk about transfusion reactions. The Rh antibody level may be undetectable for years, but produce a rapid response upon exposure to the antigen. We had a patient and he's gone, but Clyde Reeves, he came to us forever. He was a GI bleeder, unfortunately had cirrhosis of the liver due to alcoholism. But we identified an anti-Big E in his serum. He came to us for a long, long time. And then I guess he got mad at us and he went someplace else. And I guess he got mad at them and he came back. And when he came back, they didn't have his history. And his antibody had fallen below the level of detectability. I mean, we hadn't seen this antibody in years. But then he comes back and his antibody screen is positive, his direct antiglobulin test is positive. Yeah, he's having a hemolytic transfusion reaction. Antigen-negative RBC should be transfused if Rh antibodies are identified. And again, not just for this transfusion, but for the rest of their life. Hemolytic disease of the fetus and newborn. This may occur most commonly when a woman is D negative and the fetus or the infant is D positive. These antibodies may form during the first pregnancy, but usually the baby's not affected because this is the first exposure. It'll be a primary immune reaction in which the class of antibodies is IgM, which cannot cross the placenta. Maternal antibody may destroy fetal RBCs in subsequent pregnancies, and D-negative mothers who give birth to D-positive babies are given a product called Rh immune globulin, which is anti-D in a bottle that will attach to and destroy fetal cells before her system has a chance to recognize them as foreign, thereby preventing her from forming anti-D. Our next topic is concomitant Rh antibodies. I want you to know that concomitant antibodies are antibodies which often occur together. For example, sera containing anti-D may contain anti-G, and we just talked about this. Anti-G is anti-C plus D. Anti-Big C rarely occurs by itself. It's most often seen with anti-D. And anti-CE, also known as F, is often seen in combination with anti-little c. I want you to know those. I want you to know those are examples, but the most important one that you need to remember for the rest of your life is that R1-R1 individuals who make anti-Big E will frequently make anti-little c. So what we do is patients with anti-Big E should be phenotyped for the little c antigen. And if it appears that the patient is an R1-R1, then they should, if at all possible, be transfused with R1-R1 blood. This is standard practice across the United States and probably across the world because these are so commonly found together. And again, it has to be an R1R1. Anti-little c is notorious for falling below the detectable levels, and if re-exposure occurs, causing an acute hemolytic transfusion reaction, which means as the blood is going in, that hemolysis of the blood is occurring. And again, what I mean by below detectable levels, every antibody detection system has limitations, and one of them is the ability to detect lower than normal levels of the antibody. So if they have fallen below the level of detectability of this test, then you could miss it and it's not your fault. But they will have an anamnestic response which may result in acute hemolysis. We're now going to talk specifically about detection of the D antigens. There's 4 types of anti-D typing reagents available, and we're going to discuss each one. But just a quick overview, there's a high protein, which gives a faster result, but unfortunately has an increased frequency of false positives. It requires the use of an Rh control tube, but you can convert this to the weak D test. IgM is a low protein saline reactant antibody. The low protein gives fewer false negatives. It does need a longer incubation time, and it cannot be converted to weak D testing. Why? Ding, 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 ding. Because when to do a weak D test, you incubate at 37, and then you add anti-human globulin serum. What does the anti-human globulin serum detect? IgG coating the cells. What antibody class is in the low protein? IgM. So it can only be used for room temperature testing. Chemically modified is a relaxed form of IgG anti-D in a low protein medium. There are few false positives. A saline control can be performed, and it easily converts to weak D testing. And then monoclonal source, these are low protein blends of monoclonal antibodies. You need to know the preparation, the use, the advantages and limitations of each. High protein anti-D is not used as much anymore, but what it contains in the bottle is IgG anti-D. So it has IgG class antibodies against the D antigen, and it's potentiated with high proteins and other macromolecules to ensure agglutination at immediate spin. The high protein allows the cells to come together, to come closer together, which again, problem with IgGs, they're little short, tiny guys. And by adding the protein, allowing them to come into closer proximity will enhance agglutination. Unfortunately, the high protein may cause false positives with red cells that are coated with antibody. A diluent control is required. It has everything the reagent has, except it has no anti-D. And that is a control in the lab is a control in the lab. And it's a big part of what we do. And you need to know when to do them. So again, for high protein, because of the capacity for false positives, you must run the control, which has everything the reagent has, except no anti-D. False positives due to autoagglutinins. If the patient's cells are heavily coated with protein, this could cause them to spontaneously agglutinate. Abnormal serum proteins, same thing. Abnormal serum proteins can coat the patient's cells. Antibodies to the additives, which I've never ever seen. And using unwashed RBCs, which you should never do anyway, because if you're not washing them, what's in there? It's the whole purpose of washing is to remove the plasma proteins or the serum protein. So if you don't wash it, that will increase the protein content. This can be used for the weak D test because IgG class antibodies are used. IgM anti-D is a low protein saline reagent that isn't used very much and usually is used more by reference laboratories. This is prepared from predominantly IgM antibodies, so it's scarce due to the difficulty in obtaining the raw material. How do you get IgM antibodies? It's from a primary immune response. This is reserved for individuals giving false positives with high protein antiserics. So if you get a positive with a high protein and it's a false positive because your D control is also positive, then we could use this. The newer saline anti-sera require an incubation at 37. No negative controls required unless the patient's AB pos. And the reason for this is that it's a saline solution, but if you do not have a cell in the forward type giving a negative reaction, you must always run a D control. And this is because spontaneous glutination can be a problem, and you could potentially mistype someone. So again, the negative control is not required unless the patient is AB pos. Because this is IgM, it cannot be used by slide test or the weak D test. Chemically modified is the most popular. IgG is converted to a saline agglutinin by weakening the disulfide bonds at the hinge region of the antibody molecule. This allows greater flexibility so it can increase its span. So you think of a Y, make a Y above your head, then you let your arms drop. That's what happens with the IgG. It allows it to be able to grab onto antigens on adjacent cells easier. Has stronger reactivity than IgM antibodies. It can be used for the slide test, the tube test, and the weak D test. A negative control is unnecessary unless the patient's an AB pos. And again, if you do not have a tube in the forward type giving a negative reaction, regardless of what the reverse type is, how do you know that this patient isn't spontaneously agglutinating? And that is done by running a D control. And I did. I had a stat. I was working night shift. It was from the emergency room. The patient forward typed as an AB pos, and they reverse typed as an O. Yeah, that was a trip. But I was able to determine by running the D control, because it was also 3 to 4 plus, that there was there was a problem with the forward type. But again, that is the importance of running the D control when you have an AB pos person. The last type of typing sera, which is the most routinely used, is the monoclonal anti-D, and it's prepared from a blend of monoclonal IgM and polyclonal IgG. So, because it has both IgM and IgG, it's able to react at immediate spin due to the IgM component. And if you get a negative at immediate spin and you need to do the weak D test, it has the IgG component that will react at the anti-human globulin test. It is the most frequently utilized reagent. It can be used for the tube, the slide, and the weak D test. A negative control is unnecessary unless the patient is an AB pos. And I'm going to say this for the third time because it's really, really important that you think about what's going on in your testing. If you do not have a tube in the forward type that gives a negative reaction, you must run a D control because cells can spontaneously agglutinate. The patient can appear to be an AB pos, but then when you run that D control, it may be 3 to 4 plus as well. Usually it's negative. Usually it's truly an AB pos, but you cannot call it an AB pos until you run that D control. Whenever you are running a test, we need to have the controls. And for the D typing, we don't always have to use the control. There's just certain times, especially the weak D test is number one. The diluent used has a protein concentration equaling human serum. False positives due to immunoglobulin coating of test cells occurs no more frequently than with other saline reactive anti-sera. But false positives do occur. Again, the patient will appear to be AB pos on the forward type. You have to run saline or a manufacturer's control to verify that this is truly a D pos or AB pos person. The D control must always be negative. Precautions for RH typing are the same as with any other types of procedure. You must follow the manufacturer's instructions, as testing protocols for different types of reagents from different manufacturers vary. You cannot use the indirect antiglobulin test unless it's explicitly instructed by the manufacturer. For example, for those IgM anti-Ds, you cannot do the IAT. You cannot do the weak D test. Positive and negative controls must be tested in parallel with the test RBCs. Quality control is performed daily for anti-D as well as all your reagents. The first thing I do when I hit the bench in the morning is there's a little kit for QC, and that's the first thing I do because every reagent must show that it's working every day of use. Quality control for others anti-sera is performed in parallel with the tests, since these are usually not tested each day, only when necessary. For example, you don't test people for Rh antigens every day. If the only time I will test a patient for the little e antigen is when they have an anti-little e. Because I don't use that reagent every day, you have to run the controls in parallel with your patient. Let's talk about sources of error that will result in a false positive. Contaminated reagents. Never, ever, ever remove a dropper from a typing sera and place it on the table, or never, ever hold your dropper within the tube. You always, it's a free-falling drop, hold it just a tiny bit above the rim of the tube. If you accidentally touch it to the side of the tube, then you could contaminate that reagent and cause false positives. Use of the wrong typing sera. The Rh typing seras are all the same color. They're clear. So what I do is I label my tubes with all the different typing sera names. I put the bottle in front of each of those tubes. And then before I add that drop to that specific tube, I do a double check and a triple check to make sure the name on that bottle matches the name of the tube I'm putting it in. Autoagglutinins or abnormal serum proteins that coat the cells can cause them to be very, very sticky, where when you spin them down, they stick together. One time I had a patient, just for grins, I was getting false positives, so I just put a drop of their cells in a tube all by itself. I spun it down, and it was agglutinated. And again, so that's what we mean by spontaneous agglutination. Using anti-sera in a test method other than that required by the manufacturer. It is not up to you to change a single thing in that procedure. If you want to incubate for 20 minutes instead of 10, you cannot. If you want to add 2 drops instead of 1, you cannot. This is an area that, again, all areas of the laboratory are crucial and critical, but blood bankers will go down your throat if you're not following the procedure exactly as outlined. And there's nothing that annoys me more is a student that's not reading the instructions. What do I do next? What do I do next? Well, where are you in the procedure? I don't know. Well, how do you expect me to help you? Put a straight edge, put a piece of paper under that step, then push it down to go to the next step, to where when you're first doing this, you always know where you're at. I don't mind answering clarification questions, but when you're asking me what to do next when it's right there in the procedure next to you, that makes me a tiny bit grumpy. Let's talk about false negatives. Use of the wrong anti-serum. You were supposed to put in, anti-big E, and you put in anti-little c, and yeah, they were negative. Failure to add anti-serum to the test. I just talked about this. Add your serums first, each and every time. Look in your tubes and make sure every tube has serum or anti-serum. Incorrect cell suspension. If it's too heavy, then you'll get a false negative due to post-zone. And if it's too light, it's really hard to see the button. So you could use your agglutination viewer and see it, but again, that could result in a false negative as well. If you shake the tube too hard, one thing I always demonstrate to my students, especially with ABO, is I shake it off, and there you go, there's that 4+ clump for anti-A. I said, now beat it to death. Shake it, shake it, shake it. And yes, you can actually make a positive go to a negative. Reagent deterioration. This can only be detected by doing daily QC. Failure of an anti-serum to react with a variant antigen. This really is not your fault, but it's something that you just need to be aware of, that when you do additional testing and things don't make sense, then maybe that's what you have. You have some type of variant antigen. Antiserum, in which the antibody is directed against a compound antigen. This is often a problem with anti-Big C. We were donated a lot of anti-Big C because it was contaminated. I'm like, what am I going to do with this? It's not going to act like we need it to act. In summary, the Rh system is second in importance to ABO and transfusion medicine. Correct interpretation of D is essential to prevent immunization of D negative individuals, especially women of childbearing age, which may result in hemolytic disease of the fetus and newborn. It is the most polymorphic of all the blood group systems. Of the 5 Rh antigens discussed, only D typing is required. The Rh system continues to grow. Again, it's a very, very complex blood group system. The last decade has led to an abundance of information detailing the genetic diversity of the Rh locus. It has exceeded all estimates predicted by serology. Well over 100 Rhd and more than 50 different Rhce have been documented. New alleles are still being discovered. So this concludes the presentation. This was a very heavy presentation with a lot of important material. So again, same song, 100th verse. Spend time with your textbook. Spend time with this presentation. Listen to pieces of it over again. Go to the Blood Bank Guy website and listen to some of his presentations, because it's crucial that you have a basic understanding of everything we've discussed today.